Mohamed Teleb Ismail, Associate Professor, Department of Pharmaceutical Chemistry, Alexandria University, Faculty of Pharmacy, Alexandria University. Okay. I'm talking about exploiting symmetric triazines for tailoring non-hydroxamate-based matrix metalloproteinase inhibitors capable of ferroptosis induction in colorectal cancer cells. From senses, computational, and biological evaluation points of view. Colon cancer is the second leading cause of cancer-related death in the US. On average, 1 in every 20 persons have colorectal cancer. Most of the patients are above 50s with family history. The estimates of WHO showed that we are expecting more than 3 million cases in the next decade. So taking into account this global burden of colorectal cancer, great efforts are continuously going to find new treatment options. Ferroptosis modulation is one of the most important rising therapeutic approaches for battling colorectal cancer and the reversal of chemoresistance. Ferroptosis is a unique modality of non-apoptotic cell death triggered by iron-dependent phospholipid peroxidation and regulated by multiple metabolic pathways, mainly the glutathione pathway, the glutathione peroxidase 4 regulated pathway, iron metabolism, and lipid metabolism pathway. Indeed, glutathione biosynthesis is our main target. So if we can deplete glutathione biosynthesis, either by targeting its upstream glutamate 15 anti-porter system or its downstream glutathione peroxidase enzyme, which is the catalytic enzyme converting lipid peroxides to lipid alcohols. So if we could deplete this pathway, we can induce ferroptosis and cancer cell death. So what we have, ferroptosis is used in combinations with other treatments. So we have too many options to include ferroptosis, the effects of ferroptosis with other anti-cancer therapeutic approaches. But in this case, we have at least two different chemical entities with heterogeneous pharmacokinetic and pharmacodynamic properties. So what we have, an alternative approach is to use multi-target directed ligands. So we try to merge the pharmacophoric features of ferroptosis inducers with other anti-cancer agents in single multi-target hybrid scaffold so that we can have a uniform distribution and synergistic biological activity. Our group noticed that the effects of ferroptosis in targeting cancer is seldom studied in tandem with inhibition of matrix metalloproteinases. Matrix metalloproteinases are zinc-dependent endopeptidases that are mainly involved in each stage of cancer progression and metastasis. So we had a challenge to tailor this hybrid scaffold. Starting from the non-hydroxamate model of matrix metalloproteinase inhibitors, we tried to install the pharmacophoric features that lead to ferroptosis induction, which is mainly electrophilic warheads. So we used the core as triazine, symmetric triazine, inspired by altritamine, which is an FDA-approved ferroptosis inducer, and used the model of non-hydroxamate matrix metalloproteinase inhibitors to install the pharmacophoric features of ferroptosis inducers. So we got these two nice hybrid features or hybrid scaffolds containing the electrophilic warhead as alpha beta unsaturated carbonyl compound acting as Michael acceptor. Then we install another electrophilic warhead in the alpha position, which is cyano group, so that this scaffold is now dual Michael acceptor. And we started our journey for synthesis, starting from dimethoxychlorotriazine by nucleophilic substitution with probargyl amine. The N-propine intermediate was cyclized using azide compounds, click reaction, then condensed with different aldehydes to get the alpha beta unsaturated products. The other series use the same starting compound, but we used our new customized azide reagent. So the preliminary MTT assay on normal clonocytes and HCT116, the colorectal cancer cells, we found fantastic results. We see that we have two compounds exceeding doxorubicin activity. So we use these compounds as our hits for further biological evaluation on our first target, which is matrix metalloproteinase. And we got another hit. So one of these two compounds exceeded the activity of the reference. It was superior to the reference matrix metalloproteinase inhibitor. So we continued our progress in ferroptosis studies, and we found that this compound could deplete glutathione in the cancer cells. So it induces lipid peroxidation, which is the main mechanism of ferroptosis inducers, and it inhibited the growth of colorectal cancer cells. Docking studies presumed the binding features of these compounds in its molecular targets. So the molecule, our hit molecule, could bind with the active site of matrix metalloproteinase 10 and matrix metalloproteinase 13. These are two different isoforms of matrix metalloproteinases. It also could interact covalently with the glutathione peroxidase active site, which is the major target. So in conclusion, we tried to tailor a new hybrid scaffold that can simultaneously inhibit matrix metalloproteinase and undergo ferroptosis induction. Together, it inhibited the growth of colorectal cancer cells at sub-micromolar concentration, IC50 sub-micromolar. It was superior to the standard doxorubicin therapy with an acceptable safety profile. This work was published by our group in bioorganic chemistry maybe 6 or 7 months ago. It was titled Battling Colorectal Cancer via Symmetric Triazine-Based Matrix Metalloproteinase Inhibitors armed with electrophilic warheads for ferroptosis induction, and it was the first in-class dual-acting agents. I'd like to acknowledge or to thank the research team, Christine Moros, Dr. Shireen Khattab, our mentor, Dr. Nisreen Heba, Dr. Rafiq Basili, Dr. Marwa Abu Sri'a, and myself. Thank you very much.